For day 10 homework, here's a sponge body plan showing the base, water coming in, going out, and then different cell types indicated by this person. You can freeze to look at them, along with some comments on what they're for, the outer layer, and then down here, they've shown what a section would look like with an outer layer for sponges that have basement membrane. Here it is, tight junctions between cells, some of these other cell types. There's some other examples here of ways to show the same thing that all work. So this one, and you can freeze to look at it in a little more detail. Here's another one, again showing the whole body plan and then a close-up. And yet another one, the whole body plan and a close-up. This one I included. This is up to you. This is more text than I would normally put in. It takes a lot of time to put in. But if it helps you learn, then it's fine. For me, I would have done just these here and maybe some brief notes on what they're for, especially if they're related to what my sketch looks like. So I probably wouldn't need to label that as an oocyte or egg, as long as that's what my oocytes or eggs always look like in my sketches. Number 2 was placozoan tablet animal structure and function. Here's one that would be pretty fast to draw. And it shows some of these different types. One thing I wanted was a little bit more on function for some of these. So here's one that I like a little bit better. These were, one above was just fine. But here's a mucus secreting cell. There's its mucus. These are gland cells is what they were labeled. Here's a gravity detecting cell. Here's a digestive cell, which I think is meaning enzyme secretion, although I would have preferred to find vesicles of enzymes being released. And then ciliated cells. Here's another one that again shows the gland cells. Great to include enzymes here. All of these were fine. For the exam, I would expect you to know what these different cell types do. For number 3, hydrostatic skeleton, showing arrangements of circular muscles going in a circle, or you can consider them ring muscles. If that helps to not be confused about their shape, so they're extended this way. And then longitudinal muscles, we're looking down on them, and so they look round in cross-section, but they're actually running up and down out of the image. These are the ring or circular muscles. Here they are contracted, showing the fluid being forced up for extension. This person also noted that the circular muscles would need to relax to retract. When they're retracting, the longitudinal muscles shown here are shortened. So these are shortened, so they have become shorter and fatter. And that's going to pull down the top and, of course, make it also wider because there's still fluid in the cavity inside. And then to bend, on one side, here's longitudinal muscle. It is shortening, and that's going to bend a tentacle in that direction. Here's some other examples. One here and here, nicely showing the same things. And another one, again, nicely showing the same things. This one has more cells drawn than are probably needed, but if it helps you remember, that's fine. The next one was the process of forming a calcium skeleton. So this says it's seawater here. Actually, these are normally in some compartment, so they're underneath the animal, and it's forming the skeleton underneath, because if this was seawater here, all these substances could leave. So I wanted to point this one out because it's an easy misconception that it's not seawater. It's something underneath where the cells are laying down a thicker and thicker skeleton. I like this one because this person numbered the process. Here's the protein being secreted. This is the scaffold protein. Calciums can bind to it really well, and they're spaced perfectly well. This is the calcium release. This shows it from a vesicle, more likely calcium pumps. Calcium's not normally released from vesicles, although there's no reason it can't be. But calcium pumps, then the calcium binds, carbonate forms, and here showing the dark calciums and the red carbonates. And here they are forming. Here's the HCO3 minus. What's missing in this one is the transporter to take HCO3 minus from the cell and get it outside, but then here's the H plus being moved back in. So this has one missing step about where does this bicarbonate ion HCO3 minus come from. And then they've shown the whole point of this, the protein strands mixed with crystal strands. So the crystals grow from these proteins, and there are bands of protein. And what that means is when a crack forms, it runs into this flexible layer of protein and it doesn't tend to spread. Whereas without the protein layers mixed in with the crystals, once a crack forms, it tends to go all the way through. Cracks propagate easily until they run into something that stops them. Here's another example. This one didn't number it, but here's the protein coming out to the calcium. 3, the bicarbonate ion should be just HCO3 minus. Then protons being pumped in. Here's the CO3 minus 2. And then here it is growing on the crystal, the calciums, and carbonate. And just these 4 shows nicely what the process would be. This also shows it expanding. Here's another one showing the same thing. All these were good. Number 6 was complex. How do we get some signal, which might be touch or a chemical? This person did a chemical. And then all the steps to get a message down to other cells to either contract or to release glue. This person used our familiar G protein coupled receptor, cyclic AMP formed, binding to, in this case, a transcription factor, but could also bind to a channel. In this case, transcription to make the stuff that goes into a vesicle, although we could have used the cyclic AMP just to release a vesicle that has the neurotransmitter or the chemical messenger in it already. So cyclic AMP to open a calcium channel, and then the calcium causing release of the neurotransmitter vesicle, which we've talked about briefly. We'll come back to talk more about it. And here is process of calcium entry. So we've gone from cyclic AMP to calcium entry. And I went through this in class, a wave of calcium through the cell. And then at a later step, we have calcium coming in, calcium binding to a calcium binding protein associated with the vesicle. Here's the vesicle contents being released. There they are. And that might be either a chemical that binds to this person labeled muscle. Sponges don't have muscle, but it would be actin and myosin rich cells. And the cells then contract. Again, not true muscle cells. On an exam, I might take off, I don't know, half a point or something like that, because sponges don't have true tissues. They don't have muscle. So I'd take off a tiny amount for that, but this one is a really nice answer in other ways. And then here's our sponge contracting away. Here's another example shown with some steps missing. The sodium comes in. It doesn't show the sodium opening the calcium channel. And especially for homework, if you know steps, I'm pretty sure this person knows that this is the step that opens the calcium channel. If you know those steps, you don't necessarily need to show every one. Here's our calcium coming in, signal released. And then the signal either over here, G protein coupled receptor, and that's causing crystal formation. That would be toughening up the body, or it might, in this case, do microtubule disassembly protein, which would shorten the cell in this way, which was not the answer I was after, but it would work. And so I would accept that answer on the exam because it would cause the cells to contract, to pull away, and shorten the sponge. Here's another example with all the steps labeled, starting with a touch up here and going through all the stages, including producing the compounds. Here is our chemical messenger finally. So we're going here, calcium also going here, producing the chemical message. Here's the message binding to another cell. That might be at the base of the sponge and causing release of, in this case, quinones. These would be the quinones that are highly reactive and will covalently bond with 2 strands of mucus and toughen it up and make the glue harder. This was optional question 7, just practice showing how glue works. And this person showed the glue either with a quinone, this ring structure, carbon ring, covalently bonded to 2 strands of mucus to toughen it, or minus charges on an amino acid here in this strand, and a calcium that forms to by attraction of charge to stiffen it. And the last one was another optional one. Here is our simplified version of a pneumaticist, GAGs and polyglutamic acids. When triggered, water enters in really quickly. That creates water pressure, which pushes the point up. And we wind up with this pushed out toward something with the venoms around the outside.